Hello, everyone. This lesson is on Chapter 18, Section 2 on Modern Evolutionary Classification. Today, you are going to learn about the goals of evolutionary classification, and you're going to learn about a particular kind of illustration called a cladogram that describes evolutionary classification. And finally, we'll learn about how DNA sequences are used in evolutionary classification. To do that, you're going to learn a few terms: phylogeny, clade, monophyletic group, cladogram, and derived character. Let's start with a few of these terms. So, to begin, it's worth defining the word phylogeny. Phylogeny is the evolutionary history of a lineage. And in modern biology today, when we classify organisms, we do it with the evolutionary history in mind. And to do this, we group related organisms into groupings called clades. A clade is a group that includes an ancestral species and all of its descendants. So this drawing over here on the right-hand side is a drawing of a clade, and a drawing of a clade is called a cladogram, and it reads a little bit like an upside-down family tree, which is to say that the ancestors are on the bottom and the modern descendants are at the top. Okay, so let's first think of this like a family tree and consider this tip of this particular branch you. And here on this branch is your sibling, a brother or a sister. And you're connected to each other by a common ancestor. And that common ancestor would be your mom, for instance, who herself is connected to the descendants over here by means of a grandparent. So this grandparent here produced your mom and her sibling, and your mom produced you and your sibling, and your mom's sibling, her brother, produced a son who happens to be your cousin. So here we have you, your sibling, and your cousin. Okay. Let me tell you a little bit about how we talk about the parts of such a drawing. This drawing includes branches, these long lines here, and nodes. So this node connects two branches here of two related relatives, and this node here connects two branches here that connect to more distantly related groups. The deeper you go, the more distantly related groups are, so the most recent common ancestry is shared by nodes that are higher up in this graph. So now let's talk about a phylogeny. What these branch tip represents are taxonomic groupings like species. So species 1, species 2, and species 3. And what this diagram shows is that species 1 and 2 are more closely related to each other than either is to species 3. Now, I want you to think carefully about that, because what I'm saying is something equivalent to saying that you and your sibling are more closely related to each other than either of you is to your cousin. And I want you to look at this drawing and see that you can be misled, but you should not be misled. You might be tempted to say that this branch tip here, because it's physically closer to this branch tip, might indicate closer relationship, but it does not. It is as illogical as saying that your sibling is more closely related to your cousin than you are, which is false. You and your sibling are equally related to your cousin. By the same token, species 1 and 2 are equally related to species 3. So let's look at an example. This example is from last section, and it includes the crab, the barnacle, and the limpet. And I think I told you last time that the barnacle and the limpet used to be considered grouped together because they both have these conical shells. But when you look at them anatomically and in more detail, what we know is that these two organisms, the crab and the barnacle, share much more in common than either do with the limpet. So a good evolutionary classification groups the crab and the barnacle together as each other's closest relative in this set, and each of them is equally closely related to the limpet. Remember what I said, the barnacle is not more closely related to the limpet, it is equally related to the limpet as the crab is. Okay. All right. Well, how did we group these organisms together? We used specific similarities. Remember that Linnaeus used similarities to group organisms within a classification scheme. Um, and we do the same in evolutionary classification, only we don't use any old similarity. We don't use, for instance, the conical shell. We only use characteristics that are shared in a group because they became different from the ancestor and its descendants. And we call those similarities derived characters. So these two animals, for instance, share the following derived character. This one obviously has a jointed skeleton. All of its legs have little joints in it. What's less obvious is that this one does too. In fact, you can hardly see it, but up here at the top where the shell opens, the little animal sticks out its legs as filter feeding tools to grasp food that's traveling by. And these little legs are jointed. So we know that the jointed exoskeleton of this animal is shared with this animal, and both have that, different from the limpet because they evolved it after they separated from the common ancestor. So it is a shared derived character. So here we have it. These two animals both have segmented skeletons, and we're mapping that characteristic directly onto this cladogram. So this little yellow mark here called segmentation indicates that this trait evolved after the split, after this node splits into two branches. In addition, both of these have external skeletons that surround their bodies, kind of in the same way that an insect does. Um, and they share that in common. This animal does not have an exoskeleton. And so these two share this trait in common because it evolved after they separated from the common ancestor. Now, it turns out all three of these share a characteristic, which is having little, they're very young embryonic stages called free swimming larvae, and they all share that in common, which distinguishes this whole set of three from other related organisms. So you can map these shared derived characters onto cladograms to understand the evolutionary history of organisms and the sequence of events that occurred to make the diversity of life what it is today. It's actually pretty cool to do it. Here's some more example of shared derived characters with animals that you're more familiar with. What you're looking at here is a dog and a cat, specifically a coyote and a lion. And both of them have a lot in common. Both of them have specialized teeth for slicing meat. Both of them have hair that provides insulation, and both of them have four legs. And those groupings make them have shared common ancestry. But interestingly, this cat here, the lion, has retractable claws, which is something that no dog has. I don't know if you have looked carefully at the claws of cats and dogs, but this is a plain old fact that you can look at a cat's paw and not see the claws until you squeeze its paw and then the claws come out. But that's not true with a dog. They're always out on a dog. So coyotes are different from lions in that lions share this characteristic with other cats that makes them different from dogs. All right, so let's consider this cladogram of animals you're probably familiar with. If you look over the list carefully, we see sharks and other fishes. We see amphibians. Amphibians are animals like frogs and salamanders and so on. Turtles, lizards, snakes, crocodiles, birds, and mammals. All of these share traits that put them in a single clade, and that clade is called the vertebrates. So vertebrates all have a bony vertebral column. Your spine is your vertebral column. And all of these animals have something that looks like our spine. Um, However, they're different from each other in different ways, and what you can see is that some groups share more recent common ancestry than other groups. In other words, turtles and mammals have a lot more in common than either turtles and mammals do have with amphibians. So turtles and mammals have this common ancestor, and a more deep common ancestor is this one that would include amphibians. Okay. What does this have to do with classification? Well, we name these groups. Okay, so a clade can be a named group, and we call a clade monophyletic or a name monophyletic if that name includes all the ancestors. Forgive me, I said that backwards. If it includes all the descendants of a particular ancestor. So let's look at this ancestor here at the bottom of this cladogram. This ancestor and all its descendants are the vertebrates. So the vertebrates is a monophyletic group. That said, not all named groups are monophyletic, and in a moment we'll look at an example of a named group that is not. Okay, so let's consider this group here carefully. We're going to look at groups that are clades and groups that are not by highlighting some of the members of this clade. Consider, for instance, this set: turtles, lizards, snakes, and crocodiles. Anybody who knows their scaly animals knows that we call that group the reptiles, right? So you've probably all heard the term reptiles. It's all those scaly creatures that live on land, 4 legs, they all make eggs, and so on. But here's the interesting thing. This group that we call reptiles is not a monophyletic group, and here's why. Because the ancestor of all of these reptiles is here at this node where my cursor is, but this ancestor's descendants include a group that is not a reptile. It includes birds. And if we had a name for this group that included all of these animals, then we would have a truly monophyletic group name. The name reptile, which excludes birds, is not a monophyletic group name. It's not a good evolutionary classification name. This group, if we had a name for it, which we do, would be a better named group because it would be monophyletic. Now I'm going to go one deeper node here. We have the same problem here because this ancestor has 2 sets of descendants. One is this group and one is the other, and we can't call them reptiles or mammals, and we have one name for this group. It's called the amniotes. So the name amniote is a monophyletic grouping. So now I'm going to show you a good evolutionary classification of cats and dogs that includes the word amniotes. All right, so what you see is that here is a phylogeny drawn in black of several of those vertebrate groups: amphibians, remember the frogs and salamanders, the reptiles, and the birds are not shown on this one, and then a variety of mammals, including some that have pouches, some, and then the cats and dogs. All right, so here we go. This group here that includes all of these creatures, the ancestor to all of them, and all the descendants of that ancestor is called the tetrapods. The tetrapods are the 4-legged terrestrial vertebrates that don't include fishes. This group here, which includes reptiles, birds, and mammals, is the amniotes. This group here is the mammals. And here we have the carnivores, which includes both dogs and cats. But cats are distinguishable from dogs because they have something that the dogs don't have. All right, so this is a good evolutionary classification, a good cladistic approach to the naming of groups. Here's your job for today. I want you to come back after you've seen this video and provide me a drawing based on these data, a drawing that is a cladogram that groups as the most closely related individuals those species of these 4 that share the most derived characters in common. As you do this, I want you to be careful. It doesn't count if the trait is missing. It only counts if the trait is present. And so what you want to do is group the animals together that have the most traits present in common. All right? Draw a phylogeny and send it to me by email when you've viewed this. Okay, when we do evolutionary classification, we come up with some surprises. So limpets and barnacles are one surprise. Here's another surprise. Two of these birds look a lot alike, the two that are on the left-hand side of the drawing. However, what evolutionary classification reveals is that in fact this American vulture is not as closely related to this African vulture as it is to this stork, and it doesn't look anything like this stork. Why is it that we group these together? The reason lies in their DNA. DNA is shared by organisms due to common ancestry, and you've seen this in a lab activity you've done. Organisms that have more similarities are more closely related to one another. So we can use similarities in DNA sequences as shared derived similarities and use them to build cladograms. And when we do, we come up with some interesting surprises. And one of them is shown here on the right. So though we name this animal a red panda and this animal a giant panda, in fact they are not closely related to each other. The red panda is more genetically similar to a raccoon than it is to a panda. And the giant panda is more genetically related to bears than either of these two are related to these. So DNA sequence data has led to a lot of surprises in our interpretation of old-fashioned classification and led us to revise in a modern way with modern evolutionary classification a lot of the species that Linnaeus first named. I think that's enough for today, and so we'll finish here. Remember to tune back in to give me your cladogram.